Hi everyone, this is Akshita Banarka. I'm an MSc student. This is my first ever YouTube video which I'm uploading. So hope you guys like it. Please comment on it and subscribe it. So today my topic is Agrobacterium mediated gene transfer. So in plant biotechnology and genetic engineering, transfer of genes or production of transgenic plants is of major importance because it confers characteristics like virus resistance, or pest resistance, disease resistance, high yield, etc. etc. So for these, the genes are obtained from higher wild plants which already have or which have naturally inherited these characteristics. So you transfer the genes from them to the host organism, and thus the host will also have these characteristics. So how do you transfer? This is by means of vectors. You can either do it by natural means or by artificial means. For natural means, you have vectors like bacterial vectors, virus vectors, phage, etc. And for artificial means, you have gene gun method, microprojectile method, microinjection method, and electroporation method, and many other methods which I'll be explaining in the later on videos. Today I'll be talking about natural based vectors, specifically on bacterial vectors, and in that I'm concentrating on Agrobacterium mediated gene transfer methods. So, Agrobacterium is a Agrobacterium vector, that is the Ti Plasmid of the Agrobacterium can be used as a bacterial vector. So Agrobacterium, it's a bacteria which belongs to a family called Rhizobiaceae. Okay, and there are of three types of Agrobacterium. One is Agrobacterium tumefaciens, Agrobacterium rhizogenus, and Agrobacterium radiobacter. Agrobacterium tumefaciens and Agrobacterium rhizogenus are virulent strains, but Agrobacterium radiobacter is the non-virulent strain. Agrobacterium tumefaciens is responsible for inducing tumor, whereas Agrobacterium rhizogenus is responsible for inducing hairy roots, profuse root growth. Okay, how does this bacteria enter? It enters through the wound. When a plant has a wound, it enters through those wounds, and thus it gains access to those plant genomes. Okay, so let me talk about a bit on its history. Like, how did they come across this Agrobacterium? So it was accidentally found out by two scientists called Smith and Transcend. So they came up with an idea that this Agrobacterium is responsible for the tumor formation in plants, and it caused something called it was responsible for the crown gall disease. Okay, and then another scientist called Brown, he told that okay, it's not possible for the bacteria to enter into the plant cell, but it can have access only to the intercellular spaces because the bacteria is big enough. And another scientist called Zenin, he told that there is some factor within the bacteria, not the bacteria as such, but some factor within the bacteria is responsible for causing tumor, and that factor is Ti Plasmid. So Ti Plasmid has again tDNA, only this tDNA is entering into the host genome. Now let me talk about the Ti Plasmid, because this structure is very important to understand how it can act as a vector. So let me show you the structure of Ti Plasmid. So the Ti Plasmid is a 200 kilobase per plasmid. It has a Ori region, it has a virulence region, it has a tDNA region, it has a conjugative transfer region, and opine catabolism region. So Ori, as already explained, Ori, as we all know, origin of replication. It helps in the replication of the DNA within the host. When it gets replicated, obviously it reproduces. Obviously, it helps to produce more proteins, more symptoms. Okay. Then comes the virulence region, which has around 24 vir genes organized within 8 operons, which I'll be explaining later on. This anti-DNA region plays a major role in the transfer of tDNA and causing the symptoms. Then comes the tDNA region. It has a left border sequence and right border sequence. Before that, Ti plasmid is of two types. One is nopalin, the other one is octopine. Nopalin and octopine. Nopalin, it has around nopalin gene. Nopalin gene, it is like around 23 kilobase per tDNA. It has 23 kilobase per tDNA. Whereas octopine, it has two segments. One is right hand segment and left hand segment. Right hand segment is 8 kilobase per, left hand segment is 13 kilobase per. Thus, this is what makes the difference. But later, the infection and all remains the same. So, moving on to the other regions, there is a left border and right border sequence, which is 25 base pairs sequence, not 25 kilo base pair, it is 25 base pair. Then comes the auxin production and cytokine production, also known as shooty locus and rooty locus. Shooty locus, it has two genes. One is tumor morphology shoot gene 1, tumor in morphology TMS, shoot gene 2. And rooty locus has tumor morphology root gene 1. And this is responsible for the production of cytokinins like isopentyl adenosine monophosphate, whereas shooty locus 1 and 2 both codes for indole acetic acid, which is a auxin. Moving on to the cytokine product. I explained you about auxin and cytokine production region. Now coming to the opine synthesis region. Opine synthesis region, it produces opines, and this opines is catabolized by the proteins that is coded by the opine catabolism gene present within opine catabolism. Conjugative, as already mentioned, it helps in the conjugation process, horizontal gene transfer method. Now, tDNA has to be transferred into the host genome, but there are no genes as such found within the tDNA which helps to carry the tDNA or excise the tDNA from the host, from the bacteria into the host. It is nothing but the virulence region which carries the vir genes responsible for transferring the tDNA from the bacteria into the host cell. Now, moving on to the vir genes. Okay, vir genes, the virulence region, it has 24 genes. It has 24 genes. In that 24 genes, there are again they are organized within 8 operons. In what are those operons? Vir A, vir B, vir C, vir D, E, F, G, and H. These operons are 8 in number, and some are monocystonic, some are polycystonic. Vir A, vir G, and vir H are polycystonic, that is, they contain more than one genes, whereas B, C, D, E, and F are monocystonic, it carries only one gene. Now, vir A and vir G, only when they are activated, other genes get activated. So they act as regulatory genes or non-consecutive genes. No, they are not expressed all the time. They are expressed only when they are induced to express. When does this induction happens? As I already mentioned, the bacterium enters through the wound. So when there is a wound, there is production of two chemicals. One is acetosyringon, the other one is beta-acetosyringon. So this acetosyringon and beta-acetosyringon triggers the activation of vir A. Vir A gene codes for vir A protein. This protein interacts with the protein product of CHVE. This is a chromosomal gene. Okay, this chromosomal gene codes for the protein, and this is the chromosomal gene found in the bacterial chromosome, and these two interact, and then when these two interact, there is autophosphorylation of vir A. When there is autophosphorylation of vir A, the activated vir A activates vir G. Vir G is found upstream to the vir box. Vir G is found upstream to vir box, and this vir box is again upstream to other genes. So when vir G Vir-G is acting as the transcriptional inducer. When it binds to the Vir box, like your Tata box, there is activation of the other genes. There is transcription of the other genes. That is Vir-C, Vir-D, Vir-E and Vir-F and Vir-H. So what does Vir-D do now? Vir-G do now? After it has bound to it, there is activation. Then comes Vir-D into play. Vir-D has a nicking function. It nicks the tDNA 3 to 5 base pairs downstream to the 25 base pair sequence. So when it nicks the fragment that is formed, it acts as a primer. From this primer, there is synthesis of DNA, tDNA, and this tDNA is now transported into the host genome. Vir-D has other two functions as well. Vir-D has other two functions. Vir-D protects from exonuclease activity and it acts as an address tag. It helps to carry the tDNA that was produced directs it into the host genome. Then comes after Vir-D comes Vir-B1 and Vir-B11. Vir-B1 helps in conjugation, and Vir-B11 binds to ATP and it has ATPase activity, cleaves ATP or breaks down ATP, producing energy for the tDNA transfer. And then comes Vir-H. Vir-H helps in detoxification, and Vir-F helps in tDNA delivery, and it also is responsible for the symptoms. Why detoxification? Now the bacteria, when it enters into the plant genome, the plant also produces some toxins. So to detoxify them, the Vir-H gene produces some Vir-H proteins which helps in the detoxification of the toxins produced by the plant and thus helping the bacterium to survive. This is about Agrobacterium mediated gene transfer. In the next coming videos, I'll explain how Agrobacterium, the Ti Plasmid can be modified, because it has some disadvantages as well along with the advantages, which I'll be explaining, and then I'll be explaining how it was modified, how it can be modified to act as a better vector. So thank you. Hope you again like the video. Please comment on it and subscribe it as well. Thank you.